Hello everyone and welcome to the last lecture of the semester. In this lecture we'll be covering subphylum Vertebrata. Now this lecture is going to be a little different from our previous lectures. It's going to be more breadth focused rather than depth focused. So we're going to cover a variety of groups of animals and we're just going to highlight some key things about each of those groups. Additionally, some of those groups will talk about some anatomy and physiology, some key things that I think are important for you to take away from this course. But the main thing about this lecture is it's a crash course on phylum Vertebrata. So it's going to be a lot of information, but just try to remember these key differences and key characteristics from each group of animal that we're going to be talking about. So we'll start with fish, and then we'll go to amphibians, then the non-avian reptiles, then the avian reptiles, and then mammals. So let's get started. This is chapter 24 through 28 in your textbook. The first group of fish we're going to talk about belong to class Myxini, and these are our hagfish. Hagfish have no jaws, no scales, and no paired fins. They also have an eel-like body that's lined with these pore-like gill openings. And so you can see them in this picture down here in this cartoon, but you can also see those pore openings here in this picture. I'm going to circle a few of them so you can see them. And these pores run along the entire length of the hagfish, and they are openings to the gills. They have a cranium but no vertebrae. This is a derived characteristic. So instead of having a vertebrae, they maintain their notochord, which is obviously derived because all the organisms that belong to subphylum Vertebrata would have had a vertebral column, but they've lost theirs secondarily. They have a cartilaginous skeleton, so their cranium is made of cartilage rather than bone. Their body fluids are kept in osmotic equilibrium with the seawater. And this is a unique characteristic because most vertebrates are osmoregulators, not osmoconformers. And as an osmoconformer, that means the hagfish allows the concentration of salts in their body fluids to match the concentration of salts in the seawater, whereas most vertebrates are osmoregulators and they will either excrete or retain salt depending on if they're living in a saltwater or freshwater environment. Hagfish often produce this slimy secretion as a protective mechanism. There's some pictures online if you're interested in seeing it. It's actually quite extraordinary how much slimy residue they can produce from just this one small, well, relatively small animal. And this is a really good protective mechanism protecting against predators because it makes them almost impossible to hold. They're carnivores or saprophytes, and when they consume food, they will oftentimes knot themselves into this kind of pretzel-like structure, which is what they're showing you in this picture down here. So they'll form a knot towards the more posterior end of the body, and then they'll push that knot towards the anterior region. And this provides them leverage while they're feeding, so they can be more anchored on and attached to whatever their prey is. And they undergo external fertilization. And after they hatch from their eggs or are released from their eggs, they undergo direct development rather than any type of metamorphosis. Next, we have class Petromyzontida, and these are the lampreys. Like hagfish, they have no jaws, no scales, no paired fins, and they also have an eel-like body with pore-like gill openings. However, unlike the hagfish, in lampreys, the gill openings are pretty much restricted to the anterior region of the lamprey. So in hagfish, they had these pore-like gill openings that extended the entire length. However, in lampreys, they only have a few gill openings and they're closer to the head. The lampreys have a cranium and vertebrae, but despite the fact that they have a vertebrae, they do retain some of their notochord. Lampreys as adults can either be parasitic or they just don't feed at all. And then those that are parasitic tend to leave these large gaping holes in their prey, which can be fatal if they get obviously infected. So here's a picture down here of what one of those kind of gaping wounds from a lamprey look like. And this is problematic in the Great Lakes as lampreys have become invasive there and have started to prey on a lot of the fish in those areas. So when people go out and they're fishing, they might find lampreys actually attached to the fish, or they'll find these large gaping holes in the fish, which ultimately they can't sell these fish. No one's going to buy them with a gaping hole in their sides. Also, they could be diseased from this wound. So this has a big effect on the economy in the Great Lakes. And then lampreys, they spawn in fresh water. They do undergo external fertilization, so they'll go from saltwater environments to fresh water, lay their eggs, and then release the sperm. And then after they spawn, they die. And then the larva will develop, and then they have to undergo metamorphosis and to become more like the adult form. And so we've talked about the lamprey larva in our chordate lecture, and how the lamprey larva is thought to resemble the shared common ancestor of all vertebrates. So they're very, very distinct larval form from their adult form. So they do undergo metamorphosis, unlike what we saw in the hagfish. Hagfish and lampreys were our jawless fish. Everything we talk about from this point forward have jaws. And we'll begin by looking at the jawed fish that belong to class Chondrichthyes. These are our cartilaginous fish, and they include sharks, rays, skates, and chimeras. All of these fish have a complete skeleton that's composed of cartilage that's reinforced with calcium salts. So this provides flexibility, but also structure and support to the animal. And they have a complete vertebral column, but they do maintain part of their notochord. They have another key difference between them and hagfish and lampreys, and that's the presence of paired fins. There are a variety of fins in cartilaginous fish that play roles in lift, thrust, maneuverability, and stability. You can see them really well in this picture of a great white shark. We have these well-defined pectoral fins that play a role in lift as well as maneuverability and stability. We have paired dorsal fins and pelvic fins that play a role in stability and maneuverability as well. And then the caudal fin, which is providing thrust for the animal as well as stability. And so these cartilaginous fish can do a lot of great things, but one thing they're not great at doing is being buoyant. If the sharks stop swimming, they will sink, as well as all other cartilaginous fish. They're quite dense. And so to help with this buoyancy issue, they have a very fatty liver, which is filled with this lipid called squalene. And this squalene liver helps to provide them some element of buoyancy. However, they do still sink to the ocean floor if they stop swimming. This is also problematic if they stop swimming, because this is how they're able to pull water through their mouths across their gills. So if they stop swimming, they will sink, in addition to the fact they won't be able to respire, so they'll suffocate. And then also Chondrichthyes are osmoregulators, and they use rectal glands to undergo osmoregulation. And this is unique to this class of animals. So their kidneys are good at filtering out a lot of things, but they're not really good at filtering out as much salt as they need to from the body tissues. So they have a special gland that helps to just strictly pull out excess salt from the body tissues, so they're not it's not too salty internal environment. Now, this doesn't apply to all chondrichthyes. There are some that maintain a high salt concentration within their tissues to prevent water loss, and some rays will do this so that they can keep the amount of water maintained in their tissues and they don't basically dry out. They'll just keep a whole bunch of salts in their tissues, hoping that the water from the environment will move into the tissues, because the salt concentration in the tissues is higher than that of the surrounding water. Cartilaginous fish are predators, and they have a variety of adaptations that have allowed them to be very effective predators. They have very good sense of smell as well as eyesight, and then many of them also have a variety of other organs that help them to sense potential prey in their environment. One of these additional organs is the lateral line, and the lateral line is a system of interconnected mechanoreceptors, which look, in this picture, they've drawn them something like this. And those mechanoreceptors run along the side of the animal, and then they also aggregate in the head of the animal. And these mechanoreceptors will pick up low frequency vibrations from any prey that's in the environment. And then also in sharks and other chondrichthyes as well, we have these ampules of Lorenzini. And these organs are electroreceptors, and they pick up electrical signals from nearby prey. So when you combine all of these senses together, it's no wonder that these organisms are able to effectively find prey in their environment. And so once they found the prey, they also have a variety of ways of catching that prey. One, of course, they have these powerful jaws. We all are pretty probably familiar with sharks' teeth. Sharks have very powerful jaws packed with rows and rows of these very sharp teeth. And those teeth are constantly replaced throughout the lifetime of the shark. And so whenever the teeth on the anterior region of the mouth get damaged or lost, they're replaced by teeth on the more posterior teeth. And these will continue to be replaced throughout their entire lifetime. So as long as a shark is alive, they are able to effectively catch prey. And then also we have jaws in rays and skates, but their jaws don't have those same type of very defined teeth. Instead, their teeth have been kind of fused into more of these packed plate-like structures, and you can see them here and here. They're clearly still teeth, but they're kind of more they're more of a kind of connected into a plate. And this is to help them because a lot of times these rays and skates are consuming invertebrates, and which will have like a chitin or shell. And so they need to have these plate-like mouth structures to crush their prey so they can get to the meaty insides. So that's what they're used for. And then these plates are even more well defined as actual smooth plates in chimeras instead of having these kind of teeth that are fused into plates. In addition to using teeth to catch prey, some rays use electricity. And so they're showing you in this picture here that there are these electrical organs in some rays that allow them to produce electrical pulses. And these will stun the prey, giving the ray enough time to effectively catch it. They can also use this electrical pulses to stun predators, giving them enough time to escape predation. And cartilaginous fish undergo internal fertilization, and they can either be oviparous, viviparous, or ovoviviparous, which means they lay eggs. Oviparous. Viviparous means give birth to live young, or they're ovoviviparous, which means that they will have the eggs maintained within the uterus, and then the young will hatch from the eggs that are maintained within the uterus and then leave through the birth canal. And so in sharks, we have the eggs are what we call mermaid's purses, and that's what you're seeing, these structures here. In this lower picture, they've kind of cut a window into the side of the egg so you can see the shark developing on the inside. And the mermaid's purse has these two structures that almost look like arms on them. I guess you can say four structures because there are two on both sides. And these will grab onto any vegetation or rocks or any kind of sturdy anchored structure in the environment. And that way the egg doesn't kind of float through the water column, putting them at more danger. Instead, the egg will anchor to some sort of substrate or some sort of vegetation, and then the shark will develop within, and then they will eventually leave the egg and fend for themselves. Next, we have clade osteichthyes, and this includes all the bony fish and tetrapods. And all the animals that belong to this clade have two things in common. One, they have a bony endoskeleton. And so from this point forward, no more cartilaginous skeletons. All the animals from this point forward have calcified bones instead. And they also have either lungs or a swim bladder. And there are two classes of bony fish that belong to this group. And we're going to talk about each of these in the next couple of slides. And they include Actinopterygii and Sarcopterygii. Class Actinopterygii include the ray fin fish. And these are the fish that you're probably most familiar with. Just to give you some example of fish that fall underneath this class, tuna, salmon, lionfish, goldfish, koi, they all fall underneath this class. And they're called ray fin fish because in their fins, they have these long, thin, bony projections that are connected by a thin membrane of tissue. And so you can see them if you look at this picture here. This is a cartoon of kind of what the ray fin fish fin looks like. And you can also see it very clearly in this picture of a fish, where you can see these thin, bony structures in their fins. Oh, not very good at drawing them, but there we go. You can see those bony projections that seem to ray out from a central point that's close to the body, hence ray fin fish. You can also see them very clearly in these mud skippers. You can see those rays very, very easily in their fins. And the rayfin fish actually make up the most diverse group of all living vertebrates on this planet. And they inhabit a variety of habitats, from freshwater lakes and rivers and ponds to estuaries to saltwater environments, from the Arctic to the tropics. You can find fish in a variety of different habitats all over the planet. And within this class, Actinopterygii, the vast majority of modern-day bony fish belong to a group called teleost fish. And it's important to note that all teleost fish belong to class Actinopterygii, but not all ray fin fish in class Actinopterygii are teleost fish. And this is because some ray fin fish that are alive today have characteristics that more closely resemble the shared common ancestor of all ray fin fish rather than some of these characteristics, some modified derived characteristics that teleost fish have. And so teleost fish have a couple of adaptations that have allowed them to be exceptionally successful on this planet. And one of these adaptations is the development of light, thin, and flexible scales. And this is beneficial for teleost fish because it allows them to be far more mobile. The ancestor of ray fin fish had dense, very closely packed scales that acted like a suit of armor, which is good for protecting the fish from predators. However, it's bad for mobility. Imagine if you're wearing a suit of armor, you can't move around nearly as well. And so having these thin, flexible scales allow them to be more mobile and to access different elements of the environment so they can occupy different habitats. They also have more flexible fins than many other fish, like the cartilaginous fish or even the shared common ancestors of all ray fin fish. Their fins are also heavily modified in many cases to perform certain functions. So they can have modified fins to help them with prey capture, with communication, camouflage, protection, suctioning to other organisms and to substrates, and the list goes on and on. So these modified and more flexible fins also aided in their ability to take advantage of various habitats. And finally, they also have a symmetrical tail with focused muscles in the tail. And this allows them to swim a lot faster than if they didn't have those focused muscles or symmetrical tail. We didn't talk about the caudal fin structure very much in cartilaginous fish, but in those cartilaginous fish, they oftentimes have an asymmetrical tail where the top portion of the caudal fin will be longer and thicker than the bottom portion of the caudal fin. But we don't see that in the ray fin fish. And you can take a look at the picture of the mud skipper below to see how their caudal fin is far more symmetrical. And this aids in their ability to swim much faster than a lot of other fish. Rayfin fish are osmoregulators, and they use a combination of their kidneys, gills, and feces to regulate how much salts and water are present in their tissues. And so for a freshwater fish, it's very important to make sure you're retaining salts and removing excess water. However, if it's a saltwater fish, it's important to remove excess salts and retain as much water as possible. So they can use, like I said, their kidneys, gills, and feces in combination to regulate how much salts and water is present in the tissues. Rayfin fish also have a unique organ called the pyloric ceca, which plays a role in lipid absorption and enzyme secretion. And you can see that in this cartoon picture down here as this structure. And they also have something called a swim bladder. And we'll talk about a swim bladder in a lot more detail in a couple of slides. However, just to give you a little introduction, the swim bladder is a structure or an organ that allows a rayfin fish to remain buoyant in the water column even when they stop swimming. So this is different from what we saw in Chondrichthyes, where once they stop swimming, they sink because they don't have that buoyancy. However, these rayfin fish use their swim bladder to remain neutrally buoyant in the water column, which is a good advantage for them. And then rayfin fish exhibit a lot of different feeding habits. The vast majority of them are carnivores. However, some are herbivores, particle feeders, scavengers, or even parasites. And this diversity of feeding mechanism is also one of the major reasons why they're able to inhabit a variety of habitats. They can occupy a variety of niches in each habitat that they encounter or they colonize. And then rayfin fish can be oviparous, viviparous, or ovoviviparous. They generally undergo metamorphosis, so they go from an egg to some sort of fry, and then they'll undergo development and metamorphosis and change, into the more familiar adult form. And so they'll continue to grow and grow even after they become an adult. Most fish can experience unlimited growth. They're only limited by once they get eaten or sick or die from some other event. However, their growth is not uniform throughout the year. They do undergo kind of waves of rapid growth and then kind of very slow to almost no growth and then rapid growth and then slow to no growth. And this rapid growth usually occurs when food and other conditions are just optimum, which is generally in the summer months because food is ample, the water temperature is great, everything is perfect. And then generally in the winter or the colder months, they will undergo a really drastic decrease in growth, maybe even a cessation of growth until the summer months occur again. And so this gives you almost like a ring structure in the fish's scales, which is what they're showing you here. So we go from a small juvenile fish, and then the winter comes, and then the summer comes, and they'll grow. They'll begin to grow once it gets warmer, and then same thing the next year, and the next year, and then so you end up with these kind of rings, almost like a tree rings, in the scales of a fish, which can help you actually identify how old the fish is and different conditions those years based on how much a fish grew between years. Swim bladders are present in the vast majority of bony fish, both those that belong to class Actinopterygii and those that belong to class Sarcopterygii, which we'll talk about in a couple more slides. But not all bony fish have a swim bladder, but it does provide some major advantages. We talked about buoyancy. It provides the animal with the ability to remain neutrally buoyant in the water column. So they don't have to constantly expend energy swimming around in order to survive. They can just stay still. They can just stop swimming, which can be good if you're trying to catch prey. You can be a lot more stealthy. It's good for when you're trying to avoid predators as well. If you need to just don't move, right? They won't see if you don't move. And then it's also good for them if they want to, sleep or whatever they want to do. They can expend less energy because they can just stop moving. They can just remain buoyant, and they don't have to use their muscles to remain buoyant in the water column. So how does the swim bladder work? Well, we begin with blood entering vessels near the swim bladder. And once blood enters these vessels, which are shown in red down here, they will encounter a very acidic environment as they get closer to the swim bladder. And this acidic environment is due to secretions of lactic acid from the gas gland, which is this structure here. And the high lactic acid concentration will cause the hemoglobin from the red blood cells to release their oxygen. And that oxygen will then be stored or deposited into the gas gland. And from there, the oxygen will diffuse from the gas gland into the swim bladder. And this is how the swim bladder is able to inflate. And so the more oxygen that's being released from your red blood cells and being put into the gas gland, the more oxygen that's able to be put into the swim bladder, and it will inflate. And so this is important if, let's say, the fish is at higher up in the water column, like closer to the surface. They need more air in their swim bladder in order to remain buoyant. However, let's say a fish wants to start diving. In order for them to do that, they need to decrease the amount of oxygen in their swim bladder, because if they have too much, then they'll start to float back up. And so for them to do that, they have a couple of different mechanisms. One is a more ancestral mechanism where they will release the air from their swim bladder directly out of their mouths. And so you'll have these bubbles of air that'll travel up to the surface. Or the derived way is to diffuse the oxygen from the swim bladder into a structure called the ovale here. So the oxygen will go from the swim bladder to the ovale and then be put back into the red blood cells that are passing past that region. And so that's a way that they can deflate their swim bladder as they're going deeper into the water column. And in some fish, the swim bladder, in addition to playing a role in buoyancy, it also plays a role in hearing. Another aspect of fish anatomy and physiology that I think is important for us to cover is the use of gills for respiration. And so what we're going to talk about now is referring to the rayfin fish, but this also generally applies to the cartilaginous fish and our lobefin fish, but there are some differences for those groups. So just keep that in mind. This is the general gist of how gills work. So in the rayfin fish, we have our gills contained within the pharyngeal cavity, which is shown here. And the gills in rayfin fish are covered by this piece of skin called the operculum. And this projection provides protection for the gills because they're awfully fragile, but it also allows for the flow of water in one direction, which we'll see in a second plays a really important role in proper respiration in fish. And this operculum, like I mentioned, is in rayfin fish. It's not present in the cartilaginous fish, which we saw with the sharks. They only have gill slits. So this is kind of where you start seeing a lot of the differences. However, how the gills work will be generally the same. And so also in the rayfin fish, they have these structures called gill rakers. And these gill rakers can be used by some fish for feeding purposes. They'll use these gill rakers to strain food from the water column, particularly if they're particle feeders or suspension feeders. But that's not the case for all fish. Some of them don't use these gill rakers for that purpose. And so when we're looking at the gills, the main part are these filaments. And the filaments contain these kind of plate-like structures called lamella. And the lamella are where gas exchange actually occurs. And gas exchange is facilitated by a process called counter-current exchanger system. And this system optimizes the extraction of oxygen from the water that passes by the gills. And so to understand this counter-current exchanger system, I think it's first important to understand what is concurrent flow and why that's not nearly as efficient as counter-current flow. So in concurrent flow, the water and the blood would be running in the same direction, so flowing in the same direction. So if we use this as an example, we have our water, and then we have our blood. And so what we're looking at these percentages is the percentage of oxygen that's present in the water and the percentage of oxygen that's present in the blood. If we had a concurrent flow, we would be all extracting a lot of oxygen from the water when our blood is depleted. So we're looking at this graph here. If there's 100% saturation of water in the water column, and there's 0% oxygen saturation in the blood, then oxygen is going to naturally diffuse from the water into the blood. And that'll continue to happen as the water continues to flow past the gills, because as we're getting more oxygen in our blood, we still have lower concentrations of oxygen in the blood compared to the water column. Where we reach a problem is when we get to this point here, where the concentration of oxygen in the water is the same as the concentration of oxygen in the blood. And when this happens, it's at equilibrium. Oxygen will no longer continue to be pulled from the water. And so you can only get about 50% of the oxygen available in the water extracted into the blood. However, if you use a countercurrent flow, you can pull out up to, I think it's 85% or more of the oxygen from the water as it passes by the gills. So if we're looking at this picture here, we're going to label it the same way we did before. We have our water, and then we have our blood. And so if the water is flowing in the opposite direction of the blood, then the concentration of oxygen in the water is always higher than the concentration of oxygen in the blood. So we're always going to be pulling oxygen out of the water into the blood. And so you can see that if you look at this picture here, we start let's say we start with 100% saturation in the water of oxygen. And then even if we do that, we have 90% saturation in the blood, which is still less. So we're going to keep pulling oxygen from the water into the blood. Same thing when you go on to the opposite end. If we have 5% saturation of oxygen in the water, we have 0% saturation in the blood, we're still going to be pulling oxygen from the water because oxygen wants to go from a high concentration to a low concentration. So this is why they have a counter-current exchanger system rather than a concurrent exchanger system. So this is how a lot of gills work in a general sense. Like I said, there are some differences depending on if you're looking at the ray fin fish versus the lobe fin fish versus the cartilaginous fish on like the structure and, is there an operculum, how many gills there are, all that types of stuff. But this is the general gist of how gills work. And then the vast majority of ray fin fish will allow kind of gulp water in and pass the gills. But many fish that have a high active lifestyle, like sturgeon and kind of the more pelagic fish, swordfish and things like that, that move very quickly, they need to be able to get more oxygen than just gulping water past the gills. So they use something called ram ventilation. So they'll swim with their mouths open and allow water to freely pass by the gills, and that allows them to have access to more oxygen present in the water, so they can have more metabolic activity than, let's say, a goldfish needs. Class Sarcopterygiae include the lobed fin fish. Two examples are the lungfish and coelacanths. And they're called lobed fin fish because of the structure and the appearance of their fins. Their fins are far more fleshy and more robust than what we saw in the ray fin fish. And you can look at them here and see that they are clearly very different from what we saw in the ray fin fish. And also looking at the structure as well is very different than the structure of the ray fin fish. Additionally, these fish often have lungs, gills, or both, and the potential presence of lungs as well as the structure of their fins is one reason why lobed fin fish are a key piece of the puzzle when understanding tetrapod evolution, because the ancestor of all tetrapods was thought to have the same characteristics, kind of these lobed fins, and eventually they developed lungs as well that allowed them to spend more time on land. And then also these lobe fin fish are ovoviviparous. The transition from aquatic lobe-fin fish to terrestrial tetrapod was a very slow one, and it required a lot of physiological and structural changes. And this is because there are various challenges of living on land that are not present in aquatic environments. One of these challenges is the increased amount of oxygen that's present in air than in water. And so we just talked about gills and how they work. And so you can probably see why gills are not an effective respiratory organ for terrestrial life. Instead, the lobe-fin fish had to develop other ways of undergoing respiration. So as they're going from an aquatic lobe-fin fish to terrestrial tetrapods, many of these early tetrapods developed lungs or were found ways to use their mouth or skin to undergo respiration. Those that developed lungs, the lungs are thought to have developed from modified swim bladders. And so this image here is just showing you the swim bladder in the lobefin fish, and it's also present in the rayfin fish, was later structurally changed and modified to become the lungs. In addition to making structural modifications to the swim bladder to become lungs, they also had to enhance vascularization to this organ. And this is because the swim bladder does have vessels that go to and from it. However, not all the blood in the body needs to go to the swim bladder. But this is different from lungs. When you have lungs, all of the blood needs to go to the lungs to get oxygenated at some point. So they had to enhance the amount of vessels that go to the lungs to these modified swim bladders so that they can properly oxygenate their blood before it's sent to the tissues. And enhanced vascularization was not only present in the tetrapods that developed lungs, it was also present in those that use their mouths and skin to undergo gas exchange as well. So you see a lot more vessels being located in the mouth or just below the skin in these early tetrapods. And in addition to these things, they had to also figure out a way to prevent their oxygenated blood and deoxygenated blood from mixing. This is generally not a problem for fish because they send their blood from the heart to the gills and then directly to the tissues. However, in tetrapods, their system doesn't work that way. We send blood from the heart to the lungs, back to the heart, and then to the tissues. And so this requires some sort of way to keep the oxygenated blood and deoxygenated blood from mixing. And so double circulation, we start to see that evolve in early tetrapods. And then another challenge they had to overcome was the increased pull of gravity. So when you're in an aquatic environment, water helps to support your weight, and this is also why we see some very massive aquatic animals. However, on land, you don't have that same amount of support. So they had to undergo some structural changes that allowed them to support their weight against gravity. So they developed stronger jointed limbs than their aquatic lobefin fish ancestors. And these limbs that we see in modern day tetrapods are actually homologous to the limbs of lobefin fish. And you can see those structures when you compare them in the lobefin fish in this picture and the early tetrapod. You see there's a lot of similarities. And then they also had to undergo some skeletal remodeling and skeletal strengthening to support their weight appropriately against the weight of gravity or the pull of gravity. And then they also had to develop larger and stronger muscles and then new muscles to like support the head and support the tail and various elements of the body weight. So in order for them to push themselves against the ground, against the weight of gravity, they had to enhance their muscles, the amount of muscles they have and where they're located and the strength of those muscles and the size of those muscles. And then another challenge that early tetrapods encountered was drastic fluctuations in environmental conditions. In aquatic environments, conditions do tend to fluctuate, but they're far more subtle and gradual because water is a really good buffer for temperature changes, salinity changes, etc., etc. However, in terrestrial environments, conditions can change quickly. It can go from being warm to cold. It can go from being wet to dry. So early tetrapods had to figure out ways to appropriately survive in these constantly, very quickly changing environmental conditions.